So, Bio 121, just a few moments ago, maybe last week, we began thinking about what it means to think about cellular respiration. And we've gotten to this place now where we've taken one 6-carbon glucose structure and we've now broken it down into 2 pyruvate molecules. So, if you recall, one of the things that I wanted to make sure you paid attention to is you have one 6-carbon structure, and you know that's glucose. By the time we finish glycolysis, we have 2 3-carbon pyruvate molecules, PYR. And now these, if possible, will undergo a series of oxidation steps. And in eukaryotic cells, if oxygen is present, 2 pyruvate molecules will enter the mitochondria. Remember I said when I finished the previous 7.1 presentation, what I said is if oxygen is present, we will do this. It will move to the mitochondria. If oxygen is not present, it will not. And for short periods of time, you're designed to function without oxygen. You do this all the time when you sprint or run or jump up and down 2 stairs, whatever. It makes no difference, right? But the point is going to be this. If oxygen is present, we're going to convert it to this molecule called acetyl-CoA. This is what is required for it to enter the citric acid cycle. Okay, what's going to happen is we're going to release a carbon dioxide molecule. It's going to be oxidized, and then that energy that's released into space and time is transferring an electron to nicotinamide adenine dinucleotide. Okay, and coenzyme A will be attached, and now we can actually direct what's going to happen. And this is really important because each of these modifications changes the shape of the molecule so that you end up with this thing that's called acetyl-CoA here, because here's our pyruvate. Cut off the carbon dioxide. It's actually called a decarboxylation reaction, which is an oxidation reaction, such that we now add our CoA at this sulfur group here. And what's going to happen is we have acetyl-CoA. The acetyl group is transferred such that you end up with the molecule that can now help us complete the mitochondrial responsibilities. So what are those? Okay, well, look, it's another cycle, and you're going to notice that what's going to happen is the acetyl group comes in, acetyl-CoA. We're now going to go through a series of, again, metabolic enzyme-driven reactions, which are a series of state of reactions in which citrate is oxidized, so you end up with 3 NADH pluses and 1 FADH2 molecule released, as well as 2 carbon dioxides released, and 1 GTP or ATP is produced. And we haven't talked about GTP before, but it's an energy intermediate made up of a different nucleotide. If this is adenine, this is guanine. And there are multiples of these. You can imagine that if you think about how DNA is made. ATGC, that G can be used to make guanosine triphosphate. The important point is this. We've made now, if citrate is oxidized completely, 3 NADH plus H pluses, 1 FADH2, 2 carbon dioxide molecules, and an ATP. And the final product is way over here. Oxaloacetate. And this continues to run in a cycle, bouncing around from state to state, releasing each of these molecules, as long as there's sufficient reactants present. And the way to think about this is, as long as you're breathing, as long as you're making acetyl-CoA, you're going to continue to make 3 molecules of NADH, 1 molecule of FADH2, 2 molecules of carbon dioxide, and either a GTP or an ATP. And you're like, okay, what's the big effing deal? Well, the big effing deal is, think about this. What do you exhale? You exhale carbon dioxide. So as long as your metabolic system is functioning, you release that carbon dioxide, and now this causes that continued buildup to occur. That's what we mean when we say the cycle runs continuously in the presence of a sufficient number of reactants. And that, in and of itself, should be pretty mind-boggling. So, what's going on here? So let's go back here. Sorry about that. This is a blown up piece of what I was just showing you here. Again, if it was a biochemistry class, I'd make you draw all of these out. Move from citrate to isocitrate to alpha-ketoglutarate, succinyl-CoA to succinate, fumarate, malate. Malate becomes oxaloacetate. And notice what happens. Count the carbons. Okay, you actually have acetyl-CoA, 1, 2, 3, 6, 6, 5, 4, 4, 4, 4, 4, back to 6. Okay, so in the process of doing all of this work, we're literally changing the total amount of energy inside the system. Okay, that's a really important idea. You're literally creating a waterfall. And that, if you think about it for just a moment, means you're creating potential energy that can then be converted into some lower end energy that will then allow the entire system to keep running as long as the reactants are present. Okay, so you get 4 molecules of ATP, 2 from glycolysis, 2 from the citric acid cycle, 6 molecules of carbon dioxide, 2 from oxidation of pyruvate, 4 from the citric acid cycle itself, 10 NADHs, 2 from glycolysis, 2 from oxidation of pyruvate, and 6 from the citric acid cycle, and 2 from FADH2. Okay, that's from the citric acid cycle. If glucose is completely oxidized, all possible electrons have been removed. So the point is this, is most of the energy from glucose stored in ATP, the carbon dioxide, or the electron carrier molecules. And you may not know what an electron carrier molecule is, but let's pause for a minute. That's the responsibility of NADH and FADH2. These are carrying electrons so that they can actually transfer that energy. So let's just do a little bit of math here. So you have 4 ATP, 6 carbon dioxide, okay, or 12 of these guys. So it looks like the electron carrier molecules are what's actually doing most of the work here. And it wouldn't be a lie. Now, oxidative phosphorylation actually occurs inside the inner mitochondrial membrane. And this is an artwork, Mother Nature artwork of creating a pathway of an electron transport chain such that you generate a chemiosmotic environment so that you can make ATP. This is the stuff of the imagination. If we think about how Mother Nature can create intermediates of energy that will allow life to occur, this is how she does it. And I'd say brace yourself, but maybe you understand some of it already. So what does she do? She makes a series of electron transporters that are embedded in the inner mitochondrial membrane. And what's going to happen here is those NADHs and FADH2s, look what they're doing. They're bringing the electrons and the hydrogen protons, and they're shuttling hydrogen protons across the membrane. And at the same time, they're taking electrons and they're moving electrons between the membrane. Okay? Now, when was the last time you tried to hold on to an electron? Okay, think about what an electron is. It's this little tiny negligible matter structure that's spinning around the nucleus of an atom. And what we're doing here is we're moving them in between these, what we call electron transport complexes, okay, because this is the chain where you actually have the subunits 1 through 4 with cytochrome C here. And what you're going to do is you're going to move them as a result of this. And we're not going through all the biochemical processes, but hopefully you can see the energy is being transferred. Now, what's actually happening here is hydrogen protons are going to increase their concentration on one side here, and you're going to see if this is the inner mitochondrial membrane here, and this is the intermembrane space, mitochondrial matrix, what's happened is you're going to now end up creating an ability to harness the energy inside this space. And if you've ever wondered why you actually breathe oxygen, you're actually going to learn that right now. What happens is Mother Nature takes that energy and she holds those oxygen molecules in place and she cuts them in half. Remember, oxygen is O2. You take the energy, you break those into individual oxygen molecules. Remember I told you oxygen is hellfire, and that hellfire of course means that these are incredibly reactive. But what are we doing here? what we're doing is we're taking a series of hydrogen protons, and because those hydrogen protons are such at a high concentration, you slap on a hydrogen proton here, slap on a hydrogen proton here, and lo and behold, you'll notice what you're doing. You're making metabolic water. So literally when you're breaking down materials, you're making the universal solvent to ensure that the body maintains, or at least the mitochondria maintains, the total amount of water required for this entire system to work properly. In this process, those hydrogen protons are pumped from the mitochondrial matrix such that oxygen is reduced so that it can form the H2O. Now they say it's reduced here. By reduced, what they mean is split so the hydrogen protons can actually bind to it. And that's a fairly amazing idea. So what's going to happen next then? Well, you can see what's going on here. You have these specific structures which are B vitamin derived molecules with an iron sulfur structure. They literally use a transfer system. Think of like hot potato, the game. You move the electrons in as you're passing them through. The chemiosmosis piece here, the part that you probably want to go watch, right, is now remember, we're going to take those hydrogen protons that are now moving down their concentration gradient through this thing that's called an ATP synthase. And they take that ADP, they take that energy that's coming down, the hydrogen protons moving down their gradient, and that potential energy is now used such that they can attach that phosphate to that ADP. And that's what you see here, ADP plus P plus the energy from hydrogen protons moving down their concentration gradient allows that energy to be stored as ATP. And this is how we make 90% or more of the ATP inside your cells as they're functioning. So this is what's actually happening. This is sort of summary inside the actual mitochondria itself. Remember, glycolysis occurred outside here. Citric acid cycle gives us our products, and you can see how materials are moving. Notice all the hydrogen protons are being pushed in this direction, creating a hydrogen proton gradient. So those hydrogen protons now move down their gradient to make that ATP. So this is how they get to that 30 to 36 I was telling you about before. And notice I haven't lied to you. It varies by species and how efficiently the NADH, nicotinamide adenine dinucleotide, from glycolysis enters the mitochondria. Okay? And cellular respiration in total stores about 34% of the energy from glucose in ATP. So that means 66% of the energy is being moved someplace else. And if this was more efficient, I tried to explain this to some physics students once, right? They're like, well, why isn't this 100% efficient? Well, if this was 100% efficient, what would you actually have? Go away and think about that. What would actually happen inside of your cells? Now, in the absence of oxygen, you still have metabolism, but when oxygen is lacking, fermentation generates NAD+. Okay? And otherwise, glycolysis would halt. And there are two common types of fermentation. You have lactic acid fermentation and alcohol. Lactic acid fermentation you've experienced. You've ever run suicides or whatever they call them these days, or full court sprints or cross field sprints, whatever. If you do them enough times, you end up throwing up if you've done them properly. What happens is you've changed the pH inside of your body as a result of that lactic acid fermentation, and your body needs to get rid of that pH change. Alcohol fermentation, this is what maybe you're not doing it now, or maybe you are doing it now, but this is what's collected when metabolism is going forward without oxygen, and this is the material that crosses the blood-brain barrier rather easily, such that you end up becoming intoxicated. Okay, so where are we going to go with this? So this is lactic acid fermentation here, pyruvate to NADH, lactate to NAD, and in the presence, when oxygen becomes available again, this reduced structure can now be converted back to pyruvate, which is important because when you're sprinting for your life, you can make this and survive for a short period of time. Once oxygen becomes available, you can now convert it back to this, and now you can send everything to the mitochondria for it to function properly. There are plenty of breweries around here now where you can go see how this is done, but notice fermentation tanks have valves so that the pressure inside the tanks created by carbon dioxide can be released. Otherwise, they would blow up. Right? That's really all there is to it. And here, if you want to go take a quick look at how this changes when you can measure respiration. You can measure the change in temperature, you can measure the change in pH, but ultimately what ends up happening, of course, is how you actually can tie these to the rest of metabolism. Because if that's carbohydrates, if you think about glucose, you can also use lipids and the other molecules. You can use intermediates of glucose metabolism, where you have amino acids, which can be converted into glycerol, so you can have glycolysis. Some amino acids directly can go to pyruvate oxidation. Some amino acids can be directly sent into the citric acid cycle. The point is this. Remember, lipids have a glycerol backbone. That glycerol can be converted into this system. Amino acids, you'll see, I think I might show you some of it. The citric acid cycle, this is why you need to ingest essential amino acids. And when your diet isn't healthy, that's when you actually feel sluggish. So here's a citric acid cycle, and you can see what I built for you here is these are the different amino acids that can be shunted into the citric acid cycle. Notice some of it is outside, some of it is inside. We can go to and away from glucose. If we had time to think about what that actually might mean, we would do that. But what I want you to see here is this is really, really dynamic. So even though we teach you this really simple, look, acetyl-CoA comes in and ultimately oxaloacetate in the presence of more acetyl-CoA can continue this entire system. There are multiple ways to enter the citric acid cycle. Now, how do you regulate this? Hormonal regulation is going to control glucose of entry into the cell. Put an asterisk next to that. For the purpose of this class, this is what we're going to teach you. Insulin regulates glucose entry into a cell. Diabetics, at least one type of them, lose the ability to do this, and then all sorts of hell breaks loose and you die a slow, painful death. Now, there are other ways for this to actually work, but primarily it's good enough for this class. Enzyme reversibility or irreversibility can make sure that cellular respiration is regulated, as well as enzyme sensitivity to pH changes, which is where that lactic acid buildup comes back in, and this gets us back to that feedback control system we were talking about a few classes ago. And here's a good example. Insulin allows glucose into the cell. We phosphorylate it, and then once inside the cell, we can begin to break it down. GLUT4 allows glucose to actually enter the cell, and what's going to happen is now what we do is we allow this phosphorylated structure to undergo glycolysis. So this is a very sort of straightforward mechanism that allows for insulin to bind that insulin receptor to now allow for the mechanism for glucose to actually enter in. And here's a summary of feedback controls in cellular respiration. When you read the entire chapter, you should be able to create tables like this. And you can see here glycolysis to a pyruvate to acetyl-CoA, our citric acid cycle and electron transport chain. These are the centrally regulating enzymes. And this is the effector that is acted on by those enzymes. And if so, this is what would happen to the pathway. And that would be the way you would read it. The electron transport chain, in the presence of high amounts of ATP, will have the pathway slowed down. You don't need it, so therefore you don't make it. Whereas over here, phosphofructose kinase, when you make fructose 6-phosphate, it increases the pathway so you can accelerate it all the way through to the formation of pyruvate and then ultimately acetyl-CoA. So we've worked through chapter 7, and I can't stress for you greatly enough how important this is. But remember that this is what actually happens when we take one 6-carbon structure and ultimately we break it down into 2 3-carbon structures, and then we process that through the citric acid cycle and the electron transport chain so that ultimately we get this. And that's that ATP. And remember, when we cleave off that gamma phosphate, that's what releases energy to drive most, if not all, of the metabolic, remember, anabolic and catabolic reactions inside all of your cells, inside the entirety of your body, but also throughout all living organisms. Think blue whale to, I don't know, woodchuck. Okay? Bye.